Something I clearly left out of the syllabus, and I don't know how. I think I did this last semester too, when I made sure to put that review of concepts that we were all going to be together and you guys were going to do the review. I think I just slipped this out and thought, wow, everything worked out perfectly. So we're going to be, I think we're going to be one chapter behind when we get out of this first exam. And so what I left out was this pathways of vision, really getting that information beyond the visual cortex. And that's what I started in the last slideshow with the visual cortex and beyond, because I did start talking about the where, or whether you want to call it the where or the how or action pathway. And in the literature, just so you know, you will see different researchers call it different things. And as Ungerleider and Mishkin showed, so we have these two pathways, the one for the where, how, taking action, but then also through the temporal lobes, that ventral pathway for the figuring out of what, the object recognition pathway. And so I have this beyond feature detectors because I think it's pretty interesting the way the visual system pulls this information apart into little tiny dots, basically, in the retinal ganglion cells and in the lateral geniculate nucleus with those center surround receptive fields. We put those dots back together into features, vertical lines, horizontal lines, moving oblique lines. But we want to perceive the world, right? We want to perceive the table and the desk and our dogs and our cats and our friends the way we perceive objects in the environment. So we need to get beyond. We need to put those features that are back in the visual cortex still pulled apart in those hypercolumns, but representing objects back there in these hypercolumns with features. We want to put those features back together and see real objects. So some of the people who discovered where and how this is done, we started with Charles Gross and colleagues in 1972. And this is another one of these really interesting serendipitous results where they were, so if you look at the stimulus number 1 on the left, they were starting with dots, just like Hubel and Wiesel saw in the retinal ganglion cells and in the lateral geniculate nucleus. And just like Hubel and Wiesel were starting with in the visual cortex. And even though they had really figured out, hey, we're getting into features here, as we got into the temporal lobes, they were still saying, well, we're going to start with dots. And what they were seeing was there was no response of the neurons. And then in this day when the monkey was still hooked up and the experimenter was just pointing at something and the shadow of the person's hand got on the projector screen, that's when they saw that particular neuron fire. So they got excited, they changed their materials, and you can see that as we move into where I have 2s and the 3s and the 4, as we move into looking a little more like we could have finger-like projections, but we're still not finger-like, but at least it's not just a circle or a square, but we have these ridges and so forth. There's some neural firing. As we get into, well, the 4s and the 5s, as the 4s really start a little bit to look like hands, or the second 4 really looks like a hand, the 5 is clearly a hand, we see more firing. The maximal firing, once they got their stimuli together, was from what looks like a monkey's paw, so a monkey hand. And so we are seeing neurons in the inferotemporal cortex of the monkey responding to more complex stimuli, and these particular neurons were responding to hands. And just to show another study later on, as they continued to find these neurons that were responding to more complex stimuli, Tanaka et al. In a couple of studies found cells that respond to, I always call them the lollipop cells. They respond to what looks like to me to be a lollipop. And now on the bottom of what you see in A, that is neural fire, the amount of neural firing. And so that spike up, that neuron is really firing to that lollipop. If we give it just the stick, not so much firing. If we give it just the circle, again, not so much firing. If we give it a lollipop that is a square instead of a circle, it's firing, it has a pretty strong firing rate, but if you can really tell in the slide, if you do this kind of with your hands and go across, it's not quite as strong as with the circle, with the lollipop. So I don't know if they called this a lollipop neuron, but I do in my teaching. So as we get into the temporal lobes of the brain, and we're doing in this what pathway, the cells are beyond feature detectors. They're responding to complex stimuli. And we have particular regions in the brain where those neurons are responding best to similar complex stimuli. And they call these structures that are specialized to process information about a particular type of stimulus a module. And I'm going to walk through some of these modules that have been discovered, but at this point, there have been there are lots of different complex stimuli that we have discovered where exactly we are processing, like things like tools and animals, and it's really kind of neat. It leads to this argument or this way of talking about neural firing and brain representation as a modularity of processing, because we do have these modules in the brain. But I'm going to continue on talking about this a bit as distributed processing, that we have distributed processing across the brain for a particular complex stimulus. We still do have these modules where we see a greater activity of a particular area for a particular type of stimulus. So I have just a few slides here talking about neurons that respond to faces. These were discovered in the monkey's inferotemporal cortex, or the IT cortex, where those neurons were responding best to pictures of faces. So anything along the top, whether they were human or monkey faces, we see response, which is shown with the green bar, and the firing rate. And then if we're showing them other pictures of buildings or flowers or non-face pictures, we just don't see. We see almost no neural firing. I would not call that much inhibition, but maybe even a teeny bit of inhibition. This was from Rolls and Tovey in 1995. Tsao et al. In 2006 did another study where they started talking about this fusiform face area, but really for the monkeys, this was the inferotemporal cortex. This was in their temporal lobes. They were giving them, so it was a bit larger of a region for monkeys, where they were giving them pictures of faces. They also took those non-face pictures and broke them into categories, and you can see that there's really not much neural response for any specific category, whether it's bodies, fruits, gadgets, hands, or scrambled. Really, those neurons are responding to faces. And then also in 1997, Nancy Kanwisher and her colleagues, using fMRI, found the fusiform face area in humans. And you can see in this picture that inferotemporal cortex is that area that we're talking about in the monkeys is pretty large. And for humans, when we're talking about the fusiform face area specifically, that is this small area. That's actually a brain turned upside down, where that is, if you want to point to your right side, right under your temple there, if you kind of go up through that way, if you were to stick a recording device up through that way far enough, you would get to your fusiform face area, where we have neurons that are responding best to faces. And we knew that we had this module, or this region of the brain, that was responding to faces from neuropsychological evidence, even before studying monkeys or people, that we knew something about this, because there are people who have this specific kind of brain damage. It is not too common, and I was looking for something on YouTube to explain and talk about prosopagnosia. I usually tell a couple stories here, but I'm gonna find a short YouTube video. I think I found one, hopefully, where there's damage to the temporal lobe, specifically the fusiform face area, and it causes a difficulty in recognizing the faces of even very familiar people sometimes. So depending on the severity, I think the people I've seen so far in this video, they recognize a couple of faces in these pictures. Some people have it to the point of not recognizing their family, not recognizing themselves in a mirror. It can be really severe. It just depends on the amount of damage. And this is additional evidence that this area, this fusiform face area, is specialized for the perception of faces.